Okay, good morning, everybody. How are y'all doing today? All right. So today's my favorite day of the year. It's the BioChallenge 2025. We have folks from all across the country and all across the world here this morning to share in this pitch competition whereby we're handing out $100,000 in cash to five amazing neuroscience companies. And we're privileged to have you here in the room with us. My name's Chris Khalil. I'm the executive director of the New Orleans Bioinnovation Center. And I'm currently trying to make sure our slides work, and they do. So I'm thankful for our AV team in the back, making sure that that happens. So I've been with the Bioinnovation Center now 13 years, 5 years of my capacity as executive director, and now 13 years of my capacity as managing director of our seed fund, the New Orleans Bio Fund. At Bioinnovation Center, we help bold scientists and entrepreneurs turn breakthroughs into real world impact. We're more than just a building. We're a launch pad for biotech innovation across Louisiana, providing lab space, investment capital, and mentorship that founders need to grow. Over the past decade, our companies have raised over $500 million and created over 1,000 jobs, all right here in New Orleans. And if you believe in the power of science to change lives, we want you to come along for the journey with us. I want to thank our program sponsors for today. The University of North Texas Health Institute for Translational Research, led by Dr. Sid O'Brien, who is our keynote speaker this morning. The Biodistrict New Orleans, JP Morgan Chase, Jones Walker, Answer ALS, LED LAIO, DDD, GNO Inc., VentureWell, Entergy New Orleans, Delta Utilities, LSU Health Foundation, Home Bank, Advanced Office Systems, BCM, Genhart, and more. Thank you very much to our team who worked tirelessly to make this happen. Thank you to our planning committee and to our semifinalist judges who helped narrow down 110 applications down to 5. To our finalist judges who have a very difficult task of naming a champion today. To our NOBIC board members, investors, guests, and last but not least, our 5 startup finalists from across the world. Everybody, welcome to BioChallenge 2025. While you're here, don't forget, please connect with us on socials. Feel free to take pictures with our giant brain leased to us by the Tulane University Brain Health Institute. Take a selfie, tag at New Orleans Bio, BioChallenge 2025. Also, our glass wall is a place where lots of folks like to take pictures and tag on socials. So please help us amplify this by doing so. This is being live streamed on YouTube. It'll be on my permanent record and the permanent record of all of the startups here this morning. So let's blast this far and wide because we've migrated this competition from a local post-Katrina competition focused on insular local universities to something that's global. And we're still supporting our local ecosystem by making sure that those companies have every opportunity to participate and to be strong to compete. But we've made this a global pitch competition with the intention of bringing companies to New Orleans and anchoring them here. We've raised a good amount of cash from many of the folks here in this room. We're incredibly thankful for your support. And we're handing out $100K free and clear to companies this morning with no questions asked. Our goal is to integrate these companies into health systems as well as build investment syndicates around them. We're giving away $50,000 to first place, $25,000 to second place, $15,000 to third, and then the folks here in the audience are going to have a chance to vote on their audience favorite, and they'll be receiving $10,000. Okay, a little bit about the Bioinnovation Center. We have beautiful wet labs here available. We have Class A office space available. And as I mentioned, we're providing important support to companies matriculating along their milestone pathway. So we'd love for you to come and join our community. Those of you on YouTube verse, please come down to New Orleans. Our house is always open for you, and our community will welcome you with open arms. We had an amazing group of neuroscience experts and leaders to help us embark on this vision to bring the best brain health and neuroscience companies to New Orleans and to work hard and integrating them into our local community. And we're incredibly thankful for those that gave us their time once a month over the last year to bring this to reality. So let's give a round of applause to our 2025 Planning Committee. Want to talk about kind of where we've come. So the reason we're putting on the Bio Challenge is because that's how NOBIC is able to put our mission on a global stage. As I mentioned, we're handing out $100,000 this year as we did last year, and we're building our pipeline of investors, clinicians, and partners. Since last November, our 2024 finalists together have raised over $12.5 million in venture capital and about $3 million in non-dilutive capital. This fall, last year's winner in Bermuda just opened its clinical lab at the Bioinnovation Center and began pilots with Ochsner Health. Again, our goal is that these companies here have every opportunity to anchor their businesses here in New Orleans. We chose brain health because the science is finally ready to meet patients, and the unmet need is absolutely massive. You'll hear a little bit more about the importance of focusing on brain health from our keynote speaker in just a moment. We had an incredible representation of applicants this year, and we're incredibly excited about what's to come in the future as we're now on the global stage as a biotech pitch competition hosted in New Orleans with global reach. We had 110 companies from 15 countries and 22 US states apply, and we picked the 5 best to represent the Bio Challenge here in New Orleans. We had an amazing group of semifinalist judges to help narrow down the field from 110 to 70, then down to 20 and down to 5. Let's please give a round of applause to our semifinalist judges. Thank you very much. Couldn't do it without you guys. And these are the 5 finalists: NEURO BIONICS, CENTRON BIO, JuneBrain, ORIGAMI Therapeutics, and SCULPTA. We're incredibly proud, incredibly honored to have you here. Thank you very much for giving us your time, your consideration, and we hope that you give us every opportunity to convince you to do some business here in New Orleans. All right, with that, I want to bring up the gentleman with whom we could not do this without. Dr. Sid O'Brien and I had a very brief conversation earlier this year about what we're trying to do with BioChallenge, and he got it immediately. Kind of without thinking about it, he said, "What do you need? What can I do to jump in and support?" And as an LSU guy, he was ready to come back home and see how he can jump in and help support our BioChallenge. He gave us the confidence to focus on neuroscience and brain health and to do more than just hand out a couple checks. We want to build policy initiatives behind this, and we want to punch above our weight as a small 501c3 in downtown New Orleans. So thank you, Sid, for your inspiration, and thank you for all you do for those founders who are in brain health. Okay, so now I'm going to read Sid's blurb. So Dr. Sid O'Brien is Executive Director of the UNT Health Institute for Translational Research, where he leads the team in their mission to achieve a world of optimal brain health for all through research breakthroughs. He's one of the world's leading experts in blood-based biomarkers and a global leader in cognitive aging and brain health among communities by building study cohorts based around them. He leads multiple initiatives for the generation of precision medicine models for a range of neurodegenerative diseases, including Alzheimer's disease, Parkinson's disease, Lewy body dementia, and Down syndrome. Welcome to the stage, Dr. Sid O'Brien.
Well, good morning. How are you? Come on, y'all can do better than that. It is really an honor and a pleasure to be here. Getting the call, thanks to Phyllis. This means a lot to me. I grew up right down the street in Gonzales, Louisiana. Went to high school in Sonoma High School, undergraduate at LSU, and came back and did my residency at the New Orleans VA. The old VA. I haven't seen the new one. My office was 17 foot underwater after Katrina because, I was here for that. And so being able to have the opportunity to come back and help support and facilitate neuroscience in Louisiana is something that I'm very passionate about because there's so much opportunity here. And Louisiana, New Orleans fits a specific niche that the neuroscience global space needs and is basically empty. There's not many people doing it. And we were talking about this yesterday. So I'm going to talk about a couple of things that have laid the foundation for things we're doing that can be of service and utility to you. Basically, what I'm here to do is figure out how can I help. And so starting from the academic side, I run the largest brain aging study among underserved communities in history. And so why? Why are we doing that? Because African Americans have 2 times the risk of Alzheimer's and related dementias as compared to non-Hispanic whites, yet we do not know why. Hispanics, 1.5 times increased risk. We have this amazing new framework of the ATN framework for Alzheimer's disease, amyloid, tau, neurodegeneration, which was created specifically for clinical trials and has been really key to getting therapies to patients. The problem is 90% of the research on those biomarkers for the framework were well-educated, highly non-complex, no medical comorbidity white people. And so whether or not those biomarkers actually reflect the broader population is unknown. So we started the Health and Aging Brain Study Health Disparities, HABS HD, which at the time of funding, to give you a sense, was the single largest grant in the history of the NIA, larger than ADNI and larger than ACTC. So this is an enormous program. We've enrolled over 1,600 Hispanic, 1,600 Black, and 1,600 non-Hispanic white. This is a brain aging study, so we are 30 and above. We've captured, by this point, over 20,000 brain scans. We have MRI, amyloid, and tau PET. We have over 6,000 amyloid PET and 4,000 tau PET. So in terms of an ATN imaging characterized cohort, that is the largest collection of brain scans on the planet anywhere ever done. We have functional exams, we have cognitive assessments, whole genome sequencing, GWAS, proteomics, everything, and it's freely available. All of this data is at Lonnie. It does not matter if you're in academia or industry. Data doesn't go away. Take it, use it. We've given out over 400 databases in the last 36 months to 32 countries in almost every state in the United States. We have no ownership of this. You do not ever need to include me as an author on your publications. As a matter of fact, I'm asking that you don't, because I don't want to slow down your publications because I don't have time to read them. But we support trainees. We support grants. For those who have trainees, the HABS HD Challenge just came out, which is a extremely unique thing where early career scientists can submit and you get 5 minutes in my protocol. I'm not giving funding. I'm not giving samples. But if you have a questionnaire, a cognitive assessment, something that can fit within 5 minutes, you can put it in the parent protocol and we will run it for you for 6 to 12 months. 6 months gets you 1,000 people. We see over 2,000 people a year. So you get all of the scan data, all of the everything associated with it, and it costs nothing. So the HABS HD Challenge is ongoing right now. You got trainees, whatever, let them know. Love to see it. Okay, so now we're now, because of this and some of my frustrations with academia, building an industry-facing component. So this is another resource that can be available to many of you because my expertise as a scientist is people. I've built cohorts longitudinally followed and cohorts that other people say weren't able to be recruited. I was told you can't recruit Blacks into research. Almost 2,000 people later with brain scans, I'm telling you, yeah, you can. These communities want to be in research. Just get out of the ivory tower and go to the community and do the research. And so that's what we're doing now. We're building trial-ready cohorts, but our trial-ready cohorts are not, they're not demographic data. It's not a name in education. We are doing MRIs and amyloid scans, cognitive assessments and everything, so that if you as a company want to have access to the data, you simply put your inclusion exclusion criteria into our data portal, and you will know exactly who can be reconsented and followed up and enrolled into your research within the next couple of weeks. So we already have 5,000 people pre-consented for recontact and enrollment into that. But what is we're doing now that is definitely a little more unique in addition to the diversity is we're building a trial-ready cohort specifically out of primary care. So I've done this before, recruited 500 primary care patients specifically for this sort of work. So we're recontacting and expanding to have a primary care clinical trials network under, and we do tons of education for primary care docs. What is Alzheimer's? How do you detect it? What are some considerations? So it's about education, inspiration, and empowerment for that primary care network. That primary care network then becomes a source for us to expand and reach out. We're talking with Jeff, with Pennington, et cetera, about, hey, how do we collaborate with cognitive assessments and things like that? The whole point of this network is within the next 36 months to have a very new, innovative, fully comprehensive neurodiagnostic process in primary care. And I want to see it in Louisiana. The next piece is something that is extremely near and dear to my heart. Operation Enduring Brain Health. We're doing the same thing here. We're going to enroll over 2,000 veterans specifically into a trial-ready cohort. We're already started. As a matter of fact, Texas just gave me $5 million to start this. The Lieutenant Governor's Office, you all heard about DIPRIT. The Lieutenant Governor's Office was so excited about this, they funded us ahead of DIPRIT because they didn't want to slow down. So this is another opportunity. These are things that we're doing. So if you need biomarkers, we can collect and capture your blood samples, process them however you want it, send it to whatever lab you want. You need saliva, we can capture that. You need brain scans, we can capture that. You need trials and patients for trials, we can do that as well. So the whole point of all of this is to help. And as with primary care, this is, like I said, it's a piece. I've worked in 3 separate VAs. My namesake was a veteran. And so, it's how do we help provide more access to care to potentially one of the most underserved segments of our population in our veteran community. And so with that, I want to say we all are on a mission here. I want you to think big, be bold. I'm tired of incremental steps. I'm sick of, well, we did this. We can do better. My grandmother deserved better. Your family deserves better. A world of optimal brain health is within reach, and as Phyllis pointed out last night, in the neuroscience space, it's evolving rapidly. The biomarker space is evolving 10 times faster than the therapeutic space. So there's so much opportunity through collaboration, through community partnerships, industry partnerships, academic partnerships. We can bring a world of optimal brain health to the wonderful people of this state and to the people of this country. So I appreciate your time. You reach out to me and let me know how can I be of service to you. Thank you.
Really appreciate it. By the way, we're already committed to sponsor next year. Thank you very much. You have now.
So we already committed to sponsor next year as well. So if you haven't already, please do. Last night I heard you like, all right, we committed.
Another round of applause for Dr. Sid O'Brien. But we're incredibly honored to have our extraordinary panel of judges this year, leaders whose work is shaping the future of brain health, biotech, and patient-centered innovation. Each of these judges bring a unique perspective to the BioChallenge, representing the very best of science, entrepreneurship, and purpose-driven collaboration. But I think most importantly, their collective brains are bigger than that one that's sitting out there. And they have a really great understanding of the nuances of these startups and their place in the market. So we're really excited to have them judge. They have a very difficult task. to select the winners. And they took their time out of their day this week to meet with each of the finalists to see how they could help them individually. So please just give a round of applause to our finalist judges. And I will have the pleasure of introducing each one. You just met Dr. Sid O'Brien, and I think he deserves another round of applause. Yeah. One of the leaders in brain health, and we just found out we have an LSU guy from down home to lean on and to help support us moving forward. So thank you, Dr. Sid. Dr. Phyllis Farrell is a trailblazer in Alzheimer's innovation and one of the most influential advocates for patients and caregivers worldwide. We found her earlier this year when we built an NFL event in partnership with Tulane for the Super Bowl, and we haven't looked back. So thank you, Phyllis, for the thousands of intros you've made for us, the thousands more that you will make, and for your presence here today and last night. All right, so Dr. Jen Hart, former managing director, Ben Franklin Technology Partners, has been a very, very long-standing supporter and champion of the Bioinnovation Center and the BioFund. Jen is our investment committee chair at the BioFund and has supported us with over a decade of her time and support. Personally, she's done a lot for me in my role as a leader in the community and somebody trying to figure out how to deploy biotech capital in New Orleans. And very thankful for her support and for her presence here as well, and for her contribution to the BioChallenge prize pool this year. Thank you, Jen. Dr. Dave Weiner, a visionary in neuroscience and biotech, whose leadership has guided countless breakthroughs from concept to clinic. Dr. Weiner has been a trusted advisor to many emerging neurotech companies around the world and those here locally here in New Orleans. Dave is a New Orleans local. He sits on Tulane's Ventures Investment Committee and advises several companies in the neuroscience space. We're very much honored to have Dave here with us this morning. Round of applause for Dave Weiner, please. Christian Howell, Chief Executive Officer, Cognito Therapeutics, a Cambridge-based company pioneering non-invasive light and sound stimulation to slow Alzheimer's progression amongst other indications. Backed by over 300 million? More than that? Okay, yeah, way more than that. And FDA breakthrough device status, Cognito's approach has shown dramatic reductions in brain atrophy and cognitive decline, redefining what's possible in neurodegenerative care. His work exemplifies the intersection of neuroscience, technology, and compassion. And Christian is a Tulane alum, a proud Tulane alum, and a proud adopted son of New Orleans. Last but not least, Dr. Trivia Frazier, my dear friend and champion of Bioinnovation Center, all of NOBIC's programming. Trivia is a serial entrepreneur and visionary who's helping shape the future of regenerative medicine right here in New Orleans at the Bioinnovation Center and at the beach. Through Obitala Sciences, her company, she's championing inclusive innovation, creating tissue models that reflect the true diversity of the human body. Thank you, Trivia, and thank you for all you do for NOBIC. Round of applause for Trivia and all of our finalist judges. Okay, without further ado, let's get the show started. So let's say welcome to our first contestant, CENTRON BIO, Jaya Pearson, Leary. Here you go, sir. The floor is yours.
Sure. Morning. How's everybody doing today? So I'm not gonna be able to match Sid's amazing energy, but I absolutely share his passion for taking bold moves to try to solve some of the biggest problems in brain health today. And so I am the Chief Business Officer and co-founder of CENTRON BIO, and we're addressing disparities in brain health by developing patient-friendly genetic medicines. And that probably sounds a little bit strange, patient-friendly genetic medicines. How can this be the case? What we're doing here is we're using oligonucleotide therapeutics conjugated to brain shuttles that can be injected IV, so you don't have to do direct invasive administration of these. A lot of buzzwords there. Let's start with a little bit of an audience poll. I'd like to get to know everybody here a little bit. I do a lot of market research day to day in my day job. So how many people here have known somebody, like somebody close that's had a brain health issue, mental health, so on, myself, everybody? I mean, no matter where you go in the world, you ask this question, everybody puts up their hand. It affects everybody. How many people here have heard of oligonucleotide or ASO, siRNAs, those sort of technologies? About half of the audience. What about brain shuttles? Anybody heard of brain shuttles? more and more. You know, it's funny, every time we ask this question, we get more hands up. It really reflects how much of an interest and growth there is in this space to date. So let's talk about our unique spin on and what we're doing that's a little different. So just for everybody else, oligos are a type of genetic medicine that can change the way that Oligos are a type of genetic medicine that can change the way that we treat neurological disorders by treating disease-causing genes. Whereas traditional medicines treat symptoms, ASOs can actually treat the things that cause the disease by either upregulating or downregulating genes. This turns out to be really important because there's about 1,700 genes that have been identified to date, and that's at least the ones that are identified that can cause neurological disorders. And that's the potential for maybe millions of patients a year. If you care more about the money element, that's billions of dollars in potential revenue. And yet today, only 2 ASOs have been approved for CNS disease. And that's Spinraza for SMA and Qalsody for SOD1 ALS. If you're familiar with these diseases, they're more spinal focused than like direct CNS diseases that have deep brain manifestations. So the really fascinating thing to us about neurological disorders is that for many of them, many neurological disorders, we know what the targets are. We know the genes that cause many of these diseases. The challenge has been delivery, and that's in multiple ways. So let's talk about how industries try to solve this over the years. So the key element here for ASOs is they don't cross the blood-brain barrier, like many biologics. And so the two key ways that industries try to solve this is through intrathecal administration and brain shuttle technology, a first generation of brain shuttles. So intrathecal technology or intrathecal delivery, this is a lumbar puncture that you essentially will be getting for the rest of your life. So imagine having a kid that has a pediatric neurological disorder or something, or might be neurodevelopmental disorder. They might live a long lifespan. Every 3 months or so, maybe 6 months, they're going to be getting anesthetized and they're going to be getting a lumbar puncture. That's not patient friendly at all. Massive burden, high cost, low accessibility, also pretty poor distribution. When you talk with pediatric neurologists, we've heard this phrase in some of our market research that they become LP docs, meaning more and more often they have to this is part of their day jobs, giving these lumbar punctures. So IV ASOs would be completely transformative. So the way industries try to solve this over the years is through this first generation of TfR1 antibody shuttles. Essentially what this is, it's like an antibody drug conjugate where it recognizes a receptor on the blood-brain barrier called transferrin, and it latches onto that and pulls a drug cargo across the brain. This turns out to have better distribution, pretty good for antibody delivery, poor for ASO delivery. The thing with ASOs is that ASOs, you're trying to change the genes of these faulty genes. So you really need to get it within the nucleus of the cell. So again, we're at a delivery problem. So what we've created, so this is a second generation IV brain shuttle. The first iteration of this technology has actually already been in clinical trials. It's in the company PepGen. So this is the second generation of it to date. This is the neuropenetrant 1 platform that we developed. And this is designed specifically for delivering oligonucleotides. Instead of transferrin, we use cholesterol receptors. These are specifically optimized for getting out of the blood-brain barrier into neurons. The type of ASO we use is stereopure PMOs. This is with a pharma company that we have a partnership with. This is the safest, most durable ASO chemistry. It's been very challenging to develop drugs off of these, except for a few rare indications, because they have a very challenging time of getting these specifically into the nucleus. If you can unlock this, this is probably, in our opinion, the best ASO chemistry to use. And that gets to our technology mode. We have the only platform that can deliver uncharged ASOs, PMOs, to the brain effectively, and is capable of strong nuclear uptake. We get 10%, others get probably 0.1% uptake into the nucleus. We have a composition of matter on a library of peptides, and also we get composition of matter on the new drugs that we develop off of this platform. So just a little bit about what our platform does. Our brain shuttles are designed specifically to boost the activity of ASOs, or small linear peptides that have signals based into them that get them exactly to where they need to go in the cell, whether it's the cytosol if you're doing RNase H type thing, or you need to get into the nucleus. And so antibody shuttles, the challenge with them is really their large size obstructs the hybridization of the ASOs to its target genes. And so this is actually published data from 2 of the leading companies, Roche and Denali, in the space. You can see that they have pretty low nuclear activity relative to even a naked ASO, which is already pretty inefficient in the first place. And we actually massively boost the activity of ASOs. Let's get a little bit more into the data. So here we took a FDA-approved ASO, Spinraza, and we put it on a brain shuttle. We show we get higher brain penetration and durable CNS activity off of this. I'll go through some functional data in the next couple of slides. Here you can see on the I'm sorry. Here you can see we get strong whole brain distribution with IV injection. On the right, you can see cortical neurons showing that we get delivery to the nucleus. It's the little, if you can see the little blue cells with the dots in them, it's the nucleus of neurons. So we get strong nucleus uptake. We believe that's going to have better efficacy for patients, the potential for lower doses and safety. And here's some of the functional data. So this is showing increased SM2 protein. That's the protein that's dysfunctional in patients with SMA. So we actually increase the protein levels in the brain and the spinal cord. And we actually extend survival in the gold standard SMA mouse model for 282 days versus Biogen. Some of their initial data extends to about 50 days. So this is a very clear functional improvement in animal models. And we're never going to get to that massive goal that we have, or that bold goal of getting to that, that million plus patients that could be potentially eligible for these medicines if you have an unsafe product or an unsafe drug. So we're very, very heavily focused both in terms of the design of the peptide and the type of ASO chemistry that we're putting on it to have a very safe platform. So our peptide is optimized for chronic dosing. It's actually based off of an endogenous peptide. We took that sequence, put it into a linear peptide format. It's hypoimmune in every animal model we put in so far. We haven't seen any evidence of hepatic burn, and even at pretty high doses. And then currently we're fundraising to bring 2 programs into the clinic. Our goal is to address both de-risk monogenic diseases and also prevalent neurodegenerative diseases. Our first asset that we're developing is for a pediatric epilepsy. I worked for a long time at the Children's Hospital of Philadelphia, have a very strong passion for helping with some of these indications. This one is very clear biomarkers and good natural history data. We think a very strong plan to get this into the clinic and some clinical data by 3Q 2028. And then we have a pharma collaboration for 2 different ASO assets. This is the one that we're bringing forward. We believe they're the best-in-class MAPT ASO. So this reduces tau in the brains of patients with Alzheimer's disease. A large opportunity, potential for accelerated approval on both of these, we believe. Key risk here for the epilepsy is going to be execution. Can we really optimize our platform to show that we can get it into the brain effectively? And then for Alzheimer's disease, it's going to be biology and scale. How do we actually scale this as a small biotech company doing an Alzheimer's disease drug? We believe by the time we get first in human data, this is going to put us in a pretty good position for a pharma partnership. The pharma company we work with has a right of first negotiation on this as well at a phase 1 trial. And then finally, we have a capital efficient path to get clinical data for 2 assets. So a little bit of our traction to date. We have a commitment for a pre-seed bridge from a family office. Any additional funds in that would be helpful as well in terms of just doing some de-risking experiments or optimizing our platform a little bit further. We have potential lead investors identified for our $40 million seed round, up to a $5 million commitment from our pharma corporate venture arm. We have additional MTAs in progress. Also, we're incubated by both Bayer Co-Labs and J&J Labs. You'll see that something's unique about us is a $40 million seed will unlock a very large data package. And that's because we're based, so I'm in the US, I'm in Philadelphia, we're also up in Boston and Cambridge. Some of our companies in Australia, some's in Japan. We're eligible for an AMED grant that's a 2-to-1 non-dilutive match. Finally, we have a great team of peptide oligonucleotide chemists, a lot of really fantastic advisors, always looking for more advisors that have brought drugs to the market, especially in the Alzheimer's and rare disease space, and people that are just very passionate about this area like us. And then finally, so why now? Why Centron? So one of the things you'll probably have, many of you might have seen already, is that pharma's done a lot of preclinical deals with platform companies, brain shuttle type companies. And what's fascinating about these deals is that almost none of these companies can effectively deliver oligonucleotides. These are pretty decent at delivering enzyme replacement therapies and antibodies, but none of these platforms can deliver oligonucleotides effectively. And so we believe that we can solve this problem with the right platform, targets, team, and the plan. So today, yesterday we had a really wonderful day meeting all the panelists, the judges, many people in the ecosystem. Had a great dinner last night, met some folks from directly involved in the ecosystem here and building companies. And there's a lot going on in New Orleans. I have a strong connection back here myself. I actually went to high school at John Ehret and Marrero for 9th grade. So it's kind of surreal, honestly, being back here now 25 years later. So would love to, any strategic investors, thought leaders, oligonucleotide, peptide chemistry experts, but even beyond that, just anybody that wants to connect in the ecosystem would love to work together on this. This is a global problem. We're a global company. We think that we need to have global reach and work with as many folks as we can across the community. So thank you so much, everybody, for your time.
West Bank, what's up?
Yeah, what's up?
All right, we're going to open up the floor to our judges for 10 minutes of Q&A.
All right. Can you hear me? Yes.
Yeah.
Great job. Love the presentation and how you explained the problem. Couple questions.
Yeah, please.
How are you thinking about the market adoption and barriers to entry? If you could summarize that piece. I know you're at the earlier stages with respect to the discovery and generation, but how are you thinking about that? And then the second piece is specifically on the disparities side. You highlighted the disparities within brain health. How is your plan and approach thinking about that, addressing that problem?
Yeah, great question. So for the first one, maybe I'll talk about each product in particular. So for Alzheimer's disease, we'd probably be a third to market MAPT drug. There are a couple others there in terms of specific knockdown. So Biogen has a drug that's in development right now. They'll have a key readout next year, and there might be 1 or 2 brain shuttles are going into clinic. I think that those brain shuttles are going to have a high potential for safety concerns related to the transferrin receptor they're using for it. So anemia, that might be dose-limiting toxicity as well. So I think that even if we're a third to market drug and we get 20% market share, which is what our estimate's based off of, we think there's a room for multiple tau agents to be on the market right now, both antibodies and ASOs. It's a massive market. So there's that element of it as well. For the pediatric indication, we actually selected a pediatric indication where we know there's great biomarkers, and these kids have severe epilepsy. So they fundamentally would have to have kind of lifelong anesthesia to get intrathecal administration. The physicians that we've talked with as well said it would be extremely challenging, if not impossible, to develop an IT ASO for this indication area. So that's why we chose that one specifically. And then can you repeat the second question? It was about disparities. I just want to make sure I answer it.
Yes. How are you thinking about addressing that with respect to the highlighting of disparities within brain research?
Yeah, it's a great question. So you'll notice that a big focus of our talk is on safety and also accessibility by having an IV product. I would like to get it to be subcutaneous, but IV I think is a good starting point for it as well. One of the things we think about a lot is that to get to all those patients that are out there, especially in the community setting, you can't have a drug in these indications, or you should try to aim to have one that doesn't have REMS or black box or any of those other elements associated with it, or it's like severe safety concerns. So for us, accessibility has a lot to do with the drug design itself. By trying to develop what we think is a safe drug, it's not intrathecal, it's IV, it's something that even in a community setting, maybe someday you can do.
Thank you.
How long have you been
About a year.
that passed? What are you doing to mitigate?
Yeah, absolutely. So the ones that have, I'll give you the most common feedback that we get is that transferrin receptor antibody shuttles are clinically validated, which we fundamentally don't believe in. Right now, you have one that for Alzheimer's disease called trontinemab that's in clinical trials. It has extremely high immunogenicity. Patients have to get 10 mg of dexamethasone with every single dose of this drug. And it has, we think, high neutralizing antibodies associated with it. So I think that really there's a consolidation around transferrin antibodies. I think that's been the key element. Almost every investor we talk to is super interested in brain shuttle technologies. The way we've mitigated is by focusing on what we think we can do best, and that's delivery of PMOs that need to get into the nucleus specifically. And that's actually increased our traction pretty dramatically with investors since we pivoted our story to be around that.
I want to piggyback on those. This is Jen here. I have 3 quick questions that should be pretty easy enough to answer. One, could you make a quick comparison to PepGen and their first generation and why yours is better? And then I'll get to the second 2 questions.
Yeah, so PepGen used the cell-permeating peptide sequence that had high arginine content in it. So it non-specifically kind of interrupts the cell membrane to allow this the acids to get in there. Our founder Fazil was working in Matt Wood's lab when that was being developed. And so it was on some of that initial IP. And his big insight was like, well, what if we got rid of the arginine content and used receptor-mediated transport instead of arginine? So it's more of a specific uptake now.
Okay, got it. So when are you playing with changing up that receptor for different cell distribution? Tissue distribution?
That's a great,
models?
yeah. That's
Okay.
exactly the plan with it. Yeah.
Okay, and then second question, did you use a variety of ASOs to get that uptick in concentration in the nucleus? It's not clear how many that represents.
Yeah, we've used several different ASO
And they all
types.
kind of have that same result in
So
comparison?
anything that's uncharged, if we
Yeah.
use a charged ASO, like some of the PS backbones, we have, there's a challenge with conjugating our system to it that we have a couple of workarounds for, but we're really
Okay.
focused on PMOs.
Okay. Yeah,
Yes.
that helps delineate that. And then lastly, clinical, if you had the $40 million and it looks like you'd have 2 data packages for first in man after 3 years. So does that then include, because you have an epileptic population with status epilepticus, that you'd be able to get endpoint data soon enough? Is that?
Yeah, we believe we can get
So
within.
can you be in enough clinical centers for $40 million to get that amount of patient population with status epilepticus?
We can. I'll say so. The population we're going after is very well characterized at the Children's Hospital of Philadelphia. They have
Yeah.
a natural history cohort, and they already have what we think is enough patients, even if we enrolled at just a one center site, to be able to get
Really?
that.
Okay.
And then we'd have a key endpoint 6 months into it. It'd be EEG that's potentially usable
Yeah.
for an accelerated approval.
Yeah, I'm just trying to tease out the budget to get that in 3 years if you can really get those severe patients out of one center.
Yeah. I
Okay.
think it's a great question.
My question is just a little from a little broad state. You have a great platform technology that has a lot of opportunity, and you're already going after 2 separate indications. Are you going to be able to stay focused on that? So you have epilepsy and Alzheimer's disease. Just how are you going to sustain focus to get at least where you're at, something over the goal line before? Because other companies, if success, going to be interested in this, and I have concerns about getting too diffuse too quickly.
Yeah, it's a really good question. I mean, one of the things that we don't want to have happen, so we picked indications that we're both passionate about. I mean, Alzheimer's affects me and my family, and probably everybody has somebody that they know in that element. It just also happens to have a really good market opportunity. And then with epilepsy as well, it's the same thing. I think that it's really, one of the things we're trying to do with epilepsy at least is limit the amount of trial sites we have and have it really focused with a place that we think is going to do an excellent job at kind of coordinating the site all the way from the preclinical studies to the clinical studies as well and translating that. And then for Alzheimer's disease, I mean, it's we know that as a biotech company, you can 2 registrational trials of Alzheimer's disease would be extremely challenging to do. If the data look good, we presume that there would be an acquisition or licensing of that asset at a phase 2 trial. And that's also the right of first negotiation the pharma company has as well.
Okay. Sid asked one of my questions, which is how the heck would you commercialize this thing by yourself? And you won't, so I get that. My question for you around Alzheimer's disease is you have a platform that you could argue really targets precision medicine. Have you thought about the heterogeneity of this disease and whether you have the biomarkers that you need to potentially identify both the patients up front and then potentially response to therapy, which is a big challenge for fundraisers right now?
Yeah, that's a great question. We chatted a little about that yesterday. I've been chatting with Sid about that last night and today as well. We think with the tau product, at least we probably would want to try to enroll patients who have a low tau status. So use biomarkers to find patients we think could enroll well. What we're thinking about a lot now is prognostic markers. As you know, the field's very heavily invested in that. I'll say the pharma company that we're working with is one of the leaders in that space, and they have some machine learning models right now that are pretty good with prognostics. So we're thinking a lot about what can we do in the first in human trial where we can de-risk that and have a stratified patient population we think is most likely to respond to the treatment. That hasn't been happening in the other tau trials that we've seen to date. We think it's a unique way to approach tau. It's a good question. Cool. Thank you, everybody, very much. Appreciate it.
Thank you, Jaya with CENTRON Bio. Next, we have June Brain, Samantha Scott. Welcome to the stage.
All right. Hello, everyone. I am Dr. Samantha Scott, the CEO and founder of June Brain. I have a PhD in biomedical engineering. I focus primarily in ophthalmology and neuroimaging. I started June Brain about 8 years ago, the same year that I was diagnosed with a nervous system disorder by my eye doctor. And my vision at the time was that the eye would become a common tool to assess not just eye disease, but also neurological disease. And that's now becoming clinical practice. So at June Brain, we have now developed the world's most accessible retinal imaging device powered by AI to revolutionize brain and eye health. We're at the last 10% of our journey to market. We're now accelerating towards commercialization. We've developed a $6 million sales pipeline. We have clinically proven and de-risked the device and the AI. And we have spent years developing relationships with strategics in our space. They will serve as our commercialization partners and future acquirers. So the back of the eye, or the retina, is a physiological extension of the brain. So a technology like ours, which is also known as OCT or optical coherence tomography, can be used to monitor not just common eye diseases like glaucoma, diabetic retinopathy, but also MS, Alzheimer's, and Parkinson's disease. We have found that OCT can be predictive of neurodegeneration and also show signs of neuroprotection. So in addition, changes in the retina have been found that are associated with these diseases have been found to present up to 7 years before the onset of symptoms. And this is critical because delayed diagnosis can result in hundreds of thousands of dollars per patient in medical expenses and lost income. So while OCT is, again, a great measure of neurodegeneration, neuroprotection, it's really only located in specialty eye clinics. This is because it requires a trained technician to operate the device. They typically have a heavy upfront cost, which is a barrier to entry. They're large, usually requiring their own room in the clinic, and it's very difficult for patients with disabilities like myself to use these systems. So our solution is called the Neuro Eye. It's a wearable form of OCT where we've combined accessibility with intelligence. So unlike those conventional systems, ours is portable, so it can be used in any setting. It's automated, so it doesn't require a trained technician. It meets and now exceeds the performance of those conventional systems in terms of resolution and speed. And it's driven by a deep learning algorithm on the back end to automate analysis of our software. This algorithm specifically has been trained on over 700 patient data sets, including healthy controls and people with multiple sclerosis, and we're now expanding its capabilities into Alzheimer's and Parkinson's. But it automatically segments 8 layers of the retina. The top 2, the ganglion cell interplexiform layer, the retinal nerve fiber layer, those are typically very critical in neurodegenerative disease. So we're able to track longitudinally how those changes occur. And it's also device agnostic, so it can be used on our device as well as devices that are already existing in eye clinics. So we are working on our final commercial system now, but our MVP has been launched across clinical studies. The way it works, a patient wears the headset. It's about 1.5 pounds. Inside is a simple fixation target to help with alignment. Once the patient is aligned, we have a very simple GUI that shows a live view of a pupil camera for eye tracking, as well as a live OCT scan and vasculature in the retina. It can be used laying down at the bedside as well as sitting up, whatever is comfortable for the patient, regardless of disability. It's a simple click to acquire images, which again are high resolution, which is critical for neurodegeneration. And then it goes through our AI for automated segmentation and delivers quantitative measurements to the clinician on the spot. It takes around 5 to 10 minutes for the entire clinical exam to be completed. That's including in patients with severe disabilities. Thank you for doing the video. Sorry. Oh, next slide. So because of our design, we're able to now move OCT into these emergent adjacent markets. And the one with the most immediate demand is neurology. This is because OCT is now formally added to neurodiagnostic criteria, specifically first the McDonald diagnostic criteria for MS. Clinicians can stack diagnostic codes, CPT codes, to receive up to $300 per exam per eye, resulting in anywhere from $100,000 to $800,000 in new revenue for the neurology clinic. And again, we have designed our device for use in patients across the disability spectrum, from paralysis to movement disorders to those who are wheelchair bound. So it's well suited for neurology. And ultimately, Our goal is improving quality of life. OCT can be deployed in these settings to help with earlier detection, which again, all of you know very well, there are plenty of emerging drugs in the pipeline that will be able to treat Alzheimer's, Parkinson's in the future. Early detection will become increasingly critical. So this year we've come out from under our rock and we have been exhibiting at conferences across the country, including leading neurology conferences. We've had a lot of excitement from providers. We also had a chance to present some of our clinical data. We've now enrolled over 100 patients across 3 clinical study sites, including a local neurorehabilitation clinic. And we were able to show that we provide 25 to 50% superior image quality to the conventional, our competitor devices on the market. And we also demonstrated high usability among patients. So based on these conferences, we were able to grow our sales pipeline from 1.2 to 6 million in just 3 months. These are qualified leads, clinicians, and labs who are interested in buying our device as soon as it's available. It also includes around 640,000 in research contracts with neurotherapeutic companies who are using OCT as an endpoint in their studies. We also have pilots lined up with MedStar Health System in Maryland across 4 neurology satellite clinics and Johns Hopkins Hospital. And we've built a moat of IP protection. So we have 3 issued patents in the US, 2 owned by JuneBrain, as well as many pending in the EU and Canada, where we also have customers. So we have always involved the community in our R&D. And as a result of that, we have completed over 500 interviews with customers, from patients, clinicians, their staff, to build out our business model and repeatedly validate the need for a system. So based on this, our first go-to market is in research. We can deploy our device and our AI into clinical trials before we have FDA clearance. It will serve as a source of new data to further train our AI, as well as early market validation and early revenue. But once we have 510(k) clearance next year, we will roll out into our beachhead market, which are neurology clinics. And finally, we do have a lot of interest from the eye care community, including being able to add diagnostic measures for neurology in their care settings. So that includes optical retail stores. VisionWorks has shown a lot of interest in us. They're not able to access OCT at the moment, as well as optometry clinics. Our revenue model comes directly from customers as well. I always say they threw ours out the window and told us what to charge them, which is the best way to go. So instead of the heavy upfront cost, we have a revenue share model. So we charge $10,000 as an initial deposit, and then we'll take 20% of reimbursement that the clinic generates each month. So in this way, we can recoup the cost of one device in 5 to 7 months, and then it'll be profit for JuneBrain. And we've already confirmed with CMS and private payers that existing CPT codes cover our device. So with this revenue share model, we're able to become profitable by 2027. And again, we've been building our sales pipeline in order to meet our first year 2026 sales goal of 4.1 million. So I'm now raising a $1 million seed plus round. We've already closed and have committed $912,000, but we can oversubscribe. And we're also offering benefits. It's a strategic time for investors to join, including a 50% tax refund on your investment. In addition to bringing more investors into our round, we are specifically interested in the New Orleans ecosystem. We're always looking for clinical partners to deploy our NeuroEye into real-world settings, but also accessing data sets to make sure that our AI is trained on a diverse population. So if we win 50,000 today, it would actually have a huge impact for us. We are a very capital-efficient company. It would allow us to expand to 1 or 2 more additional clinical sites. We would love for those to be here in New Orleans to increase diversity within our studies. And our funds from our round will go specifically towards transfer to manufacturing. We already have our contractor in place, and they can scale on the market with us. And it will allow us to deploy units for our first customers. It will also help us reach our main milestones, which is 510(k) clearance by the end of next year for the device, and AI clearance early next year. Those are separate FDA submissions. And finally, again, we have developed relationships with strategics in our space. They have a history of buying companies like JuneBrain, either before FDA clearance or immediately after. We have demoed our device to their M&A teams, and they see us as the next generation of AI in order to move into these untapped markets. We have an awesome team. I'm happy to share more, as well as a very hands-on advisory board. They are KOLs in the neurology and ophthalmology space. Thank you very much. I'd love to hear questions.
Hi, Samantha. Love Louisiana in your presentation and what you'd like to do here. So that's really helpful. My question for you is about your two different use cases.
Yes.
So you said you've got a research use case basically to generate revenue prior to FDA approval where you can have a clinical use case. And so my question for you is, when you say research, what kind of research do you mean? And do you actually find that your models can be trained for research in the same way that they can be trained for clinical practice, or the cutoffs need to be different?
Yes, that's a good question. So our research use only, investigation use only customers are typically trials developing neurotherapeutics or just looking into biomarkers for neurodegenerative disease. So some of the customers in our pipeline, they have MS studies going, longitudinal Alzheimer's studies. Some of them are very small, some are with emerging pharma companies. For our AI, we are specifically looking to quantify changes in the layers of the retina. So our algorithm is great at doing that now for general neurodegeneration, remyelination, but what we would like to do is add in more predictive and diagnostic measures. So the data that comes in from these research groups will be helpful in a clinical use case as well. What's critical is longitudinal data in a diverse patient population. And for us, multiple types of OCT machines.
Hello, is it on? Yes, okay. So in following up on that question, do you then have rights to some of that data to then productize it?
Yes, so our contracts specifically do not give us ownership of the data, but it gives us the right to train our algorithms with the data. So it's de-identified when it reaches.
Right. Okay, great. A couple of other questions here. Is your sensitivity and specificity the same to conventional OCT? And you mentioned image quality being better, but you probably wouldn't want to suddenly be way more sensitive if insurers don't want to be paying for too big a capture or too much follow-on testing.
Yes, that's very true. We've heard that from insurers too.
Yeah.
So what we look at is not so much specificity and sensitivity, it's reproducibility and accuracy as well as speed.
Right. Yeah. So area under the curve can matter, but if it leads to further testing, parsing out sensitivity and specificity can be very valuable.
Sure.
And that goes to my last question, which is, have you been talking to insurers or ERISA plan managers about pre-approval for MRI? Because that's really a big sweet spot for something like this, to actually demonstrate the clinical, evidence of a disease.
Sure.
MRI is increasingly being clamped down. It's so expensive. So it's 10 times much more money than your product.
Yes, we've spoken with this at length with insurers. So when it comes to MS, for example, they're kind of leading the edge with bringing OCT into clinical practice. What we're hearing from insurers is that they would like to see OCT. It would never replace MRI, but be a between MRI objective data point. And in that way, we would have to demonstrate this with longitudinal testing, but showing that we're actually reducing disease burden by catching disease progression more quickly. So with MS, for example, what's really critical is conversion from cis clinically isolated syndrome to MS, as well as transition from relapsing, remitting MS to primary progressive MS. Those are very different burdens of treatment, different treatments.
But I mean, even for the first MRI, right?
Yes, sure. Yes, yeah. And for us, so instead of having to have multiple MRIs to track dissemination in space, OCT is now seen as a additional landmarker, the retina, that can be used to make these diagnoses.
Portable for ophthalmology uses. Can you confirm that you meet all the criteria that an ophthalmologist could use this and
Yes.
extend it?
Yes, we do. We designed it specifically to be as good as and slightly better than the typical OCT machine in an ophthalmology clinic.
I actually had reimbursement model. Have you explored how difficult those can be to enforce in certain clinical settings? I mean, tracking the revenue and reimbursement, which can sometimes have a really long delay of even 180 days, and trying to get that data monthly, you may want to explore. Maybe this is just more advice than a question, but you really may want to explore what's going to smooth that out
Yes.
most
So-
easily.
Yeah, I agree. What we'll likely do is track scan volume, which our machines track automatically and bill by scan volume. So our customers, we've asked about that alternative method because it is hard to track reimbursement, and they've given a thumbs up. for
Okay.
now. So we'll see
And
when we
last
get to
question, sorry to dominate here, all the questions, but the last one is just, let's see, what was it? Have you validated this in different positions? If somebody's laying down, you're looking at multiple retina layers, and some of those are going to be a little more engorged with blood if somebody's laying down. Have you done validation for these different positions?
So we've done different positions that are still sitting. Our pilot with Johns Hopkins is actually at the bedside. So they specifically want to use our device in patients who are laying down, and it will be a longitudinal study. So that's when we can validate that.
Fantastic
you.
presentation. Oh, sorry.
You go ahead. I'm going to text mine to Jen, so you go ahead now.
Okay, gotcha. Yeah, you have us jumping to ask questions. And I love the way you thought through the revenue sharing model. What do you see as some of the largest challenges to reaching that profitability goal that you outlined?
Yes, so it will always be adoption. Even though OCT is part of becoming more part of neurology clinical practice, it's not the go-to. There's still a level of patient or clinician education that we have to do. I mean, thankfully, in some spaces like MS, they are doing a lot of that work for us, but I think it will come down to our ability to properly train the clinic on how to use this information, what to do with it. We've made everything as simple as possible based on their feedback. So just a traffic light system, for example. Yes, there's a change, or no, there's no significant change. But I think the biggest hurdle will be education.
Thank you.
Sorry, my question was about the
sort of funding path.
Sure.
So you've raised 4.7, you're raising $1 million pre-commercial,
Yes.
but then you've got suitors around the table. Help me understand what your series is and what you need to actually,
Sure.
because when you do a usage agreement, you actually have to have a pretty high touch sales organization to make sure that the technology is being used. So I'm curious on the $1 million leads to what, and then, so maybe you can just give me a little bit of your funding path.
Yes. So we to date we've raised 4.7 million, and that's half of it has been NSF phase 1 and 2 SBIR funding, and then the other half has been dilutive funding. So our current raise, $1 million, we're very capital efficient, whether we want to be or not. But that will take us all the way to commercial launch. So that's, for example, the first 20 or so units that we can build out for customers. We already have a contract manufacturer. We're located in their space. They're ISO 1345 medical device manufacturer. So they can take care of inventory, shipping, all of that. But with these first customers, most of them are major medical systems. So our goal with that is to put one device. If they like it, it's easier to scale up within their setting. But we will have to build out a sales team. We just brought on a salesperson now, paid for by one of our VCs, which is very nice. But we'll have to build that out more for Series A.
For your Series A. So do you have a sense of the $1 million get you to what point, and then what the raise would be beyond that? So you're 1 on 10 now. What do you think the A looks like?
Yeah, so 1 million will get us through FDA clearance and first market launch. Our Series A will need to be 5 to 10 million, depending on how much revenue we generate from early sales. That will cover, we'll start raising that after we submit to the FDA. We already have Series A investors who've been following us for years, and they've made it very clear that they could possibly come in on a term sheet once we submit, and the first tranche would come after we receive clearance. But during that waiting period is when we'll start building out our sales team and raising Series A.
I see the huge opportunity in MS as well as Alzheimer's. Like Dave, there's a low-hanging fruit here with the general eye clinics, and given the health disparities in eye health that are well documented and just unaddressed across this entire country, is there a strategy to use this technology to increase access to even just eye screening for underserved populations? I mean, I see that. I think that's what Dave was getting at as well. Huge opportunity, huge market opportunity, but also just value for the community. And I just didn't hear that.
Yes, I agree. So in that follow-on market, it includes the retail optical stores. It also includes, and these are people already in our pipeline, mobile eye clinics, especially in Canada, for example, very rural. We've had a lot of traveling optometrists come to say, we have all the goodies in our bag except OCT. We can't fit a machine in our bag. So yes, I absolutely agree. I'd say a good percentage of our pipeline are more rural clinics that are they're clinicians who have moved from the urban centers, the major institutions, to start their own clinics, and they would like access to something that's a bit easier for their patient populations to get a hold of. Okay, thank you very much.
Thank you, Dr. Scott. Okay, with that, everyone, we're going to be taking a 10-minute bio break. Please help yourselves to the snacks and the food in the lobby. We have a spooky trick-or-treat station for you to come and pack a tote bag with candy and take it home. But please join us back in the lobby. Join us back in the conference room in 10 minutes. Thank you all very much. I'll take a few minutes to just talk a little bit about our trophies that we have for our contestants this afternoon. So I want to take a moment to recognize our trophy sponsor, Make Good. Noam Platt is a partner of ours at the Bioinnovation Center. His company is a design studio that blends art, architecture, and purpose-driven fabrication, predominantly for children with disabilities, and adaptive devices for those that need help and support. So look up Make Good. They're a partner of ours at NOBIC. They're located at the beach, and they do a lot of 3D printing, prosthetics for children with disabilities, and really cool things like wheelchairs for folks all across the world. So cheers to our 2025 BioChallenge trophies. Okay, now we can get started again. So with that, I'd like to introduce our next contestant, NEURO BIONICS, Dr. MJ Antonini. Round of applause for MJ.
All right. Thank you everyone for having me today. So my name is MJ Antonini. I'm the co-founder and CEO of NEURO BIONICS, where we are on a mission to expand access to neuromodulation therapy. Like many of you here, I have family members with Alzheimer's disease, depression, and substance use disorders. And I've seen firsthand how devastating those disorders can be. And I'm not the only one. Neurological diseases affect 1 in 6 people worldwide, stripping away patients' memory, mobility, and autonomy. The burden on the US healthcare system is astronomical, estimated at $1.4 trillion annually. And these diseases aren't going anywhere. Drugs are the first line of defense, but when they fail, powerful neuromodulation therapy exists, like DBS or deep brain stimulation, and they work remarkably well. Since its first approval in the 90s, DBS has seen tremendous growth with ongoing clinical trials for numerous additional conditions like Alzheimer's disease, depression, and stroke rehabilitation. The problem, however, is that getting DBS requires an invasive open brain surgery, which typically involves 2 to 3 6-hour-long surgeries while the patient is awake with a metal frame bolted to their head. This surgery is so invasive and frankly scary to patients that less than 1% of the 24 million patients that would be eligible for this therapy end up with the GBS implant. That's an abysmal market adoption. But what if there was an easier way? A way to get to the deepest part of the brain without having to open the skull? There is one. We can use the pre-existing pathways to the brain, the blood vessels. Interventional radiologists place devices like stents and coil in the brain without opening the skull all the time. They deploy using catheter and that are navigated through the blood vessel. However, those devices are typically passive and they don't have the ability to deliver neuromodulation to the brain tissue. That's where we come in. At NEURO BIONICS, we are developing a device that can be inserted through a small incision in the neck and guided through the twists and curves of the vasculature. Once deployed from a catheter, our device hugs the inner layer of the blood vessel like a stent. And from there, it delivers targeted neuromodulation therapy to the brain tissue, all without having to open the skull. To make this possible, we've developed flexible bioelectronic fibers that can record and stimulate from inside the blood vessel. They are as small as a human hair. I'm holding one right now. You might not be able to see it. That's kind of the point. Our team has been developing this technology at MIT for over a decade. We've refined it and extensively tested it in small and large animal models, which led to a number of high-profile publications and a robust IP portfolio that we've licensed exclusively from MIT. So today, advanced neuromodulation is restricted to a few dozen major centers, leaving the majority of eligible patients without access. Instead of relying on academic medical centers that are sparse all over the country, our minimally invasive therapy can be delivered at thousands of regional stroke centers, bringing cutting-edge technology to everyone around the US. Our therapy is also 3 times cheaper than traditional GPS implants. Amongst the numerous clinical indications for deep brain stimulation, we've identified stroke rehabilitation as a particularly promising first indication for several reasons. There is a huge unmet need. Stroke is the leading cause of long-term disability in the US. 800,000 patients every year suffer from a stroke, with half of them suffering from long-term disability impairments that often translate into upper arm mobility impairments. There's recent clinical trials from the Cleveland Clinic that show that if you stimulate the cerebellum while the patient does physical therapy, you can improve the outcome of the rehabilitation. Another key reason is that the vascular access to get to this target is straightforward. There's a vein going directly to the dentate nucleus. Finally, in this indication, instead of having to stimulate for 5 years, 10 years, 15 years, like for Parkinson's disease or epilepsy, we only have to stimulate for 6 to 8 months, which reduces the regulatory risk and time to market. So to double click on this, today, stroke rely on physical and patients rely on physical and occupational therapy to recover motor function. But that recovery typically plateaus after a year. With our device, the pathway changes. The device will be prescribed and implanted by the same physician that removed the clot in the patient. And then it will then deploy the device. The patient will then go home the same day and start physical therapy that will be supplemented by deep brain stimulation. That's this combination of deep brain stimulation and physical therapy that is key. The stimulation enhances plasticity, which then improves the outcome of rehabilitation. But what's really interesting is that the therapeutic window is very wide. You can go to patients 3 months that had a stroke 3 months, 6 months, even 3 years prior, and still recover additional motor function. We're following a B2B model, selling to our implantable devices to large hospitals, where the majority of neurointerventional cases are currently performed. And more importantly, we'll be able to leverage established reimbursement code. So compared to the other players in the GBS space, ours is the only technology that can do deep brain stimulation without having to open the skull. The only competitor is actually CENTRON. They also use a vascular access to get to the brain, but they focus on recording for the brain, and they are too big. They can't go to the deep brain targets. We believe the time is right for deep brain stimulation innovation. It is a rapidly growing market that is driven by our aging population and a destigmatization of mental health. And there are a number of new indications that are currently in clinical trial for GBS and that are expected to get approval in the coming years. And this market is ready to be disrupted. The technology for GBS hasn't really changed in the last 30 years. We went from 1 electrodes to 3 and to 8 with a little bit of current steering, which is not a lot of innovation in 30 years. Additionally, the same thing that happened in the cardiovascular space, where endovascular cardiology changed the whole field, is happening in the brain. It's now the 4th fastest growing medical specialty. In terms of a path to the clinical market, we'll follow class 3 medical device requiring a PMA. We'll seek breakthrough device designation after our first in-human data and expect our first product, a DBS for stroke rehabilitation, to reach market in 2031. So since we launched last year in January, we completed a design freeze and are accelerating towards the first in-human study next year. We've secured VC funding and grant funding, and we've partnered with major hospitals. We've demonstrated deployment of our devices in the brain, like here, but also across the body for phrenic nerve stimulation and pacing of the diaphragm, vagus nerve stimulation, and stellate ganglia. We've assembled a world-class team with expertise in engineering, material science, and neuroscience from MIT. As for me, I trained in medical engineering and medical physics at Harvard Medical School and at MIT. And I've spent the past decade building tools to interface with the brain and the body. We're supported by leading neurosurgeons and key opinion leaders in deep brain stimulation. We also have experienced business advisors hailing from Medtronic, Covidien, and the FDA. So today, only 1% of the eligible patient population receive DBS therapy. But that's already a $1.2 billion market with a 14% CAGR. With our technology, we're making DBS safer and simpler, substantially expanding this market and targeting the rest of the 99% of the population. Focusing on stroke rehabilitation as a first indication, this is already a $4 billion market, just in the US. But our story doesn't end there. Our fibers are a true platform technology. We can do a lot more than just recording and stimulation with electricity. We can do light delivery, light sensing, temperature sensing, pressure sensing, everything in this single fiber. So if you couple that with the fact that next to the large nerve, there's a large blood vessel providing nutrients to the nerve, that means that once we have our first product to market, we'll be well positioned to go beyond DBS and beyond the brain and target additional market in the spinal cord interfacing and the peripheral nervous system interfacing. So in January, we closed a $5 million oversubscribed seed round, which fully fund our preclinical plan. And due to our momentum and investor pool, we are selectively reopening 3 million allocation dedicated to accelerating towards our first in-human data next year. 2 million of which are already claimed by our insider, Future Ventures, and 1 million is available for investments. If we win the $50,000 prize today, we'll use that to jumpstart our platform and education pipeline, and also advance our IP package. We're also seeking introduction to the New Orleans ecosystem, investors, physicians, so interventional neuroradiologists, but also cardiac EP, and academic collaborators. So at NEURO BIONICS, we are developing a therapy with decades of precedent, deep brain stimulation. And we are deploying it through an established, minimally invasive approach. We are building the technology to make it possible. DBS without open brain surgery. So if our vision resonates with you, please reach out. I'd love to talk to you. Thank you.
So it sounds like what you're doing, and I apologize, I don't know this therapeutic area nearly as well, the stroke, but it sounds like what you're doing is disruptive to what is existing today, right? Which is a different way of delivering the deep brain stimulation. So could you explain a little bit about how that disruption is going to impact the folks that are currently doing these surgeries today, and whether you have academic and key opinion leader support for this disruption?
Yeah, that's an excellent question. So right now, all the DBS implantation are done by functional neurosurgeons. One of the major barriers to entry to this therapy, in addition to just the invasiveness, is the infrastructure. So all of those implantation are done in large academic centers, and population of functional neurosurgeons is limited. So those physicians are already overburdened and they are not seeking new patients. So one of our key opinion leaders is a GBS, like a functional neurosurgeons, and they really, really love the technology because they care for the patient. Like their workload is cannot increase further. The pool that we have is from the interventional neuroradiologist folks. That's a growing medical specialty. With, I think they are about like 5 times more a physician in interventional neurology than in functional surgery. And these folks typically have a caseload that is emergent. So someone has a stroke, they have to go to the hospital, get it removed, or there is an aneurysm that's going to burst, you have to go very quickly and call it. So they want elective cases like this one, so that they can have a more even caseload in the future.
So follow-on question, because I think you're implying this already, is right now neurology is a money loser in pretty much every hospital across the United States, because unlike orthopedics and cardiovascular, they don't actually do high revenue procedures. So is the thinking here that from a health system perspective, this actually increases the revenue source because they can do potentially lower cost procedures, but still more of them?
Yeah, that's exactly that. Like the interventional neuroradiology is an interesting specialty because you can go into that specialty while being a functional neurosurgeon or a neurosurgeon. You can also be an interventional radiologist, so on the rest of the body, and then focus on the brain. But you can also be a neurologist and then do a specialty in interventional neuroradiology. So it's a very easy way for them to get into that space. And additionally, it's just it's a lot easier to build a fluoro suite or like a cath lab rather than a whole surgical center for a theater for neurosurgery.
Well, without being crass, does the hospital make more money?
The hospital will make, the main one, the AMCs, will then have a bigger caseload because we also open access to a number of patients. Like at the end of the day, even though GBS works really, really well, there's only 1% of the patient population that receive it because of the barrier to entry. So they will have more patients. And also in other hospitals in like underreserved communities, we also, yeah, exactly.
So, sorry, go ahead.
So I think the disparities piece was a little bit addressed, though probably would like to hear more on that. Yeah. Your data on the therapeutic window of it, within 3 months, 6 months, and you even said 3 years.
Yes.
Where did that data come from? Because you're still going into the first in humans, because the therapeutic window for stroke is not typically that long.
Great question. So we're not targeting a stroke. We're targeting stroke rehabilitation. So that data is from a clinical trial from Andrew Machado at the Cleveland Clinic. So what they showed 2 years ago, and right now they are doing their phase 2 and phase 3 trial, is that by implanting a GPS directly into the cerebellum in the dentate nucleus and stimulating that region, you improve the plasticity of the brain and you improve the outcome. And they did that in patients that had a stroke 6 months, 3.
But is that your technology?
No. So they use the same mechanism, so electrical stimulation in the dentate nucleus, and we just have a different access route. Right. They have to open the skull. We go through the blood vessel.
Yeah, they're doing traditional DBS. I was just trying to understand how this directly linked to your mechanism of action and your delivery mechanism.
Yeah.
Okay. Thank you.
So my question is a bit of a loaded question. I'm going to give fair warning. Medtronic is a $30 billion company that has pioneered stroke retrieval, DBS, and catheter-based therapies. Why are they not in this space?
They actually just released a paper 2 months ago. They are exploring endovascular neuromodulation. It was in sheep. It was preclinical data, and they were going after the I think the ANT, the anther nucleus of the thalamus. So they are getting into that space. I think they got a lot of. They followed the rise of Synchron that showed that endovascular access to the brain is possible and safe even long terms, and now they are slowly getting into that space. But we have a number of patents that cover our technology. We also have a different approach with respect to the manufacturing process and the deployment. So they're getting there.
And then, sorry, just one more, which is, and I apologize. I know you guys are time crunched, so we went a little quickly through it. I'm interested by your clinical evidence path.
Yeah.
So you have a proof of concept, because breakthrough PMA is not an easy clinical pathway to go through. So you're going proof of concept to feasibility to pivotal. Help me understand what that looks like from a scope and an investment perspective.
Yeah, great question. So right now we are, so the additional raise right now we're doing at 3 million is to do a first in human outside of the US that will show proof of concepts. So it's going to be deployment of our device into the vasculature, targeting a nerve, and then we're going to show safe deployment, safe stimulation, target engagement, and safe retrieval of our technology. That's the first one. After that, we're going to follow the IDE path in the US, do early feasibility studies. So right after the first human, we're going to have to raise Series A that's going to be between 15 to 35 million, so that we can do our early feasibility studies and part of the pivotal, and then another Series B that will do the rest of the pivotal and then the commercialization. At least that's how we've been thinking about that. The number that might seem a bit low, but that's also because we're going to announce in 2 weeks at SFN a commercial partnership with a pretty well-known company that sells devices for neuroscience research, and they will resell our devices to their existing customer. That's a way for us to generate early revenue that will then be fund the clinical path.
Okay, so I'm just curious, just the investment path through. So what you expect to raise? What now? What on the? So
right now we're doing 3.
Yep.
Then it's going to be like 25 on average for the Series A. And then for the Series B, most likely like in the 60 million. That's the current thinking. To get, regulatory submission and regulatory approval. Yep.
So you focused on the technology of the device itself, the wire. Tell me about the programmable. The other end of the device, right?
Yep.
Whether you guys are working on it. The other question or concern I would have is that's also a limiting factor to DBS. So it's not only the surgery, but there are only a limited number of movement disorder neurologists that can program that and see patients. And I'm wondering, do you deal with that to give the broader access, or are you just creating another funnel point or problem?
No, that's a very good question. So right now we haven't done any internal development for the IPG. Most of the IPG out there, even for like Medtronic and Boston Scientific, they all come from the same CDMO company. So those are the Cambridge Consultant of the world and the Integer. So we're like in conversation with them and going to work with them. Our current understanding is like the limitation for the therapy is less so with the IPG, but more of the targeting of their lead into the right target. So that's why we also have a mission to have an IPG that will have like all the bells and whistles, so like rechargeable. We have a unique material for stimulating, which is a carbon-based electrodes. They have much better electrochemical properties than the classical platinum and platinum iridium, because they have a wider window that's much larger, and we can stimulate at the same current but from a lower voltage difference. So our battery pack are actually going to be smaller than traditional IPG.
I have a question about the variability of vasculature, and you could end up quite close to your target in some patients and a little bit farther. Have you done animal models to suss that out? And then, a related question is whether you need to show safety even in your initial, this $3 million budget to get to an IDE package, whether you need to show in animals at least that you don't have some flow issues that could be clot forming. Because, if you have to place a stent in there, it's already because you have to deal with a big issue. Mm-hmm. Now you're introducing something and a risk.
Yes. So the first part of your question was on, can you repeat the first part of the question?
The first was the variety of vasculature.
Oh, the variety, yeah. So that's a very good point. We are in the venous side, so it's even more variable than the arterial side. That's why there's going to be a lot of pre-surgical planning that's going to happen. So we're going to have an MRI and that will be then co-registered with like scintigraphy, like just angiography, and that's where we will be able to like screen patients where the vasculature goes to the target. And right now we've been working in like rabbits and also like pigs. We've done pretty much like an animal a month, or like 2 animals a month over the past 10 months, deploying our devices. So yes, there is going to be a lot of like planning involved in that. For the second part of the question, which was the safety. So safety has been like one of the main consideration as soon as we started developing this technology to the clinic. Right now we're using already approved like FDA-approved antithrombotic coatings. That's a polyurethane classical-based antithrombotic coatings. And we're doing our own studies internally and externally. We just sent everything to our like an external biocompatibility house to like validate all of that. The Series A will have actually 3 data packages. There's going to be the first in human that will just like acute deployment and safety and proof of concept. There's going to be a platform data set showing that we can deploy and simulate across different targets or across the body. And then there's going to be a long-term implementation with like a longer safety profile.
Great job. Fantastic. Good question.
Yep.
No worries. Cardiovascular comorbidities, right? Heart failure immediately comes to mind. And there's a stigma with the minority population in particular with respect to certain types of procedures. Opening it up to those who are qualified, that's one side. But on the other side, have you thought about that with respect to engaging with stakeholders? How would you address that from a disparity standpoint?
Yeah, I mean, that's a great question. We are at the beginning of our path. Like we've started a company a year and a half, like 2 years ago. So we haven't pushed a lot of conversation. We were engaging like patients as well, and like pretty much every stakeholder of the ecosystem. Stroke rehabilitation is one indication. We are interested in like a lot of other indications. Some of them are in the cardiovascular space as well. So there will be some exclusion criteria for therapy, and so that we won't be able to like serve everyone. But I think what we're excited about is like this opportunity of like platform technology that decrease the barrier to entry to this like neuromodulation therapy as a whole.
Thank you.
Thank you.
Thank you, MJ. Our next contestant is Origami Therapeutics, Dr. Beth Hoffman.
Hear me? Perfect. Good morning. I'm Beth Hoffman, founder and CEO of Origami Therapeutics. First, I'd like to thank the organizers and the judges for this fantastic opportunity to tell you a bit about Origami Therapeutics. At Origami, we're unfolding precision medicine to slow brain aging and extend health span. To do so, we use an innovative small molecule approach to extend brain health by focusing on the power of small molecules to alter how proteins fold. So let's dive in and let's see if I can get it to work. Oh, goody. Autophagy is the cellular pathway by which damaged proteins, DNA, organelles, and pathogens are eliminated and recycled. Deficits in autophagy lead to cellular dysfunction, inflammation, and cell death. Aging and development of age-related diseases correlate with a deficit in autophagy. So, defective autophagy is causally implicated in neurodegeneration as demonstrated by the numerous genes and gene mutations shown here in Alzheimer's, Parkinson's, ALS, and Huntington's disease. We're testing our innovative platform first on Huntington's disease, a genetic disease of accelerated aging, which will enable us to select patients and to test our hypothesis with quicker readouts. The mutant Huntington protein misfolds, becomes toxic, and blocks autophagy at multiple points in the pathway. As a result, we know what an effective drug should do: eliminate the mutated protein. We know who to recruit and what to measure so clinical trials are feasible. Huntington's disease, or HD, is an orphan disease with about 200,000 affected individuals, and a third-party projected TAM of greater than $9 billion. HD treatment options are grim indeed. There are no disease-modifying treatments. The disease strikes at the prime of life with an average age of onset of 35 years. Affected individuals are can expect a protracted, progressive decline in movement, cognition, and behavioral symptoms, and ultimately are bedridden and institutionalized. Despite these prospects, recent studies suggest that HD disease progression can be modified and that Huntingtin lowering is a clinically validated target. Oops. Too heavy a finger. We are pioneering a groundbreaking approach to slow, halt, and reverse disease progression by targeting the underlying cause of disease, the toxic misfolded proteins, via protein degradation through selective autophagy. We take a phenotype-first, human-first approach in which we screen for clinically relevant effects that we want to see in patients. We use primary human models to ensure translation to the clinic. As such, our precision medicine approach represents a paradigm shift in drug discovery. Our solution is ORI-003, a potential first-in-class, best-in-class, disease-modifying treatment for HD. 003 is a mutant Huntingtin protein degrader that reverses autophagy deficits and normalizes gene and protein expression in patient-derived HD models. Our lead chemical series is selective for mutant Huntingtin, as shown in the top graph, sparing normal Huntingtin in order to restore normal cell function. Patient-derived human neurons display unusual clustering, as shown by the blue cell nuclei indicated by the yellow arrow. ORI molecules reverse this effect and restore morphology to near normal. ORI-003 has excellent drug characteristics with greater than 85% oral bioavailability and excellent distribution in the brain, as shown in the top graph, with a brain-to-plasma ratio of 0.8. In the bottom graph, 003 dose-dependently eliminates toxic mutant protein and is well tolerated after 2 weeks of daily dosing in vivo in a mouse model. Our next steps are to perform non-GLP tox, apply for orphan designation, and move into IND-enabling studies. Our HD assets are differentiated first-in-class, best-in-class protein degraders. Among the advantages are that we directly target and eliminate disease-causing toxic protein while sparing the normal protein in order to restore cell health. Because we have a small molecule, we can treat the whole brain and body, and this also enables us to use blood biomarkers to monitor both disease progression and drug response. We work by a novel mechanism which restores critical autophagy lysosome pathway. And because we have an oral pill, or we project that we'll have an oral pill, this will be the least invasive approach to eliminating mutant Huntingtin. And importantly, the ability to have an oral pill democratizes access, which is unlike our competitors, which for the most part require in-hospital surgery or infusions. So Origami's clinical competitors all target the messenger RNA, which means that they prevent the generation of both mutant and normal Huntingtin. This approach does not eliminate existing Huntingtin protein and its aggregates and has potential to create a loss of function by eliminating the normal Huntingtin. While some competitors display some of the positive attributes, ORI-003 combines all of the preferred features. So we have potential for expedited regulatory and clinical path via orphan, breakthrough, and fast track designations. As I've already talked about, clinical trials are feasible And since we treat both the brain and the body, we have blood biomarkers to assess proof of mechanism, efficacy, and secondary efficacy measures, which are important in understanding responders and non-responders. Commercial opportunity is projected to be greater than $9 billion by 2030, with reimbursement anticipated for demonstration of disease modification. I should add here that all assets have been discovered and are wholly owned by Origami. So we've raised almost $7 million to date from founders, family, and friends, but also we're part of Y Combinator and have been funded by Catalytic Impact Foundation, Princeton Alumni Angels, and New Fund Ventures. In addition, we've received $2 million of non-dilutive funding from 2 SBIR Phase 1 awards, as well as 2 Kickstarter awards from O2H Discovery, which has helped us with our chemistry. More recently, we've signed a compound testing agreement with the HD-focused CHDI Foundation. So we're seeking $5 million seed to advance our HD asset to IND approval and to advance our tau degrader for Alzheimer's disease-related dementias to development candidate. So on the top, you see in purple milestones and timeline for Huntington's disease, below the timeline in green milestones for Alzheimer's disease-related dementias. So once we get to IND approval with our seed funding, we'll be looking at roughly $40 million Series A in order to complete a Phase 1 trial in healthy volunteers and HD patients where we can demonstrate clinical safety as well as proof of mechanism and dose range finding. Potential exits are really frankly the usual at this point: licensing of assets, M&A, and IPO. So the use of the $5 million goes to greater than 80% R&D, the usual activities as a course to get to IND approval in first in man. I'm joined by a foundational leadership team and advisors with more than with more than 100 years of success in pharma and biotech. I myself have put more than 30 compounds in the clinic with 5 marketed drugs for cystic fibrosis and pain. I'm very excited about being here because I see opportunities to collaborate with the NOLA ecosystem, which includes preclinical biomarker discovery and translation to the clinic, enhanced disease biology understanding using our tool compounds, and potential clinical trial sites. So I've told you who we are and what we do. Why do we think we're going to win? We're targeting the underlying cause of disease. We have unique positioning with limited competition in a growing market of $9 billion for HD, a strong initial traction, and the ability to scale to additional indications as demonstrated by our second indication, tau degraders. So we welcome you to join us in creating a world without neurodegeneration. Thank you.
Okay, so there's a big difference between a genetic form of Huntington's disease and sporadic Alzheimer's disease. And so my question for you is, from a probability of technical success, how do you compare and contrast those two indications?
Yeah, so that's, thank you. So the reason that we go after monogenic forms of neurodegeneration are exactly to try to, basically to de-risk and increase the probability of success. And by that, I mean, if we know who to recruit into the clinical trials based on say an autosomal dominant mutation as in HD, as in some primary tauopathies, as in FTD tau, we have the opportunity to actually be successful. And even though that's a smaller population than all of AD, to me, it seems, important for us to have some success. And as a company, obviously, but as a field in general, in order to bring, I think, more interest and funding into the field to tackle some of the more challenging issues of the cases in AD where we don't really know what the absolute cause is.
So your ADRD Nilex is actually targeting monogenic forms.
Initially, yes.
Good.
So
Got it.
what I think could it work in AD at large? It's possible. We don't have that data yet. So it's early days for us on that score. Yeah, absolutely.
So I understand the targeting of monogenic, but with some of these repeat diseases, they can get quite long. So even within that one disease, you have very different phenotype, including what it binds. It pulls other proteins that also have repeats into its aggregates.
Yes.
So my question here is, how much have you done in the way of animal studies to look at whether you end up with too much cellular debris, because that can be its own worst enemy, like overly efficacious efforts to use the heat shock protein pathways, for example, years ago found, too much debris, or like antibody therapeutics caused encephalopathy, too much debris. Can you talk about, how you deal with a disease that could have like a 220 repeat sequence or a 35 repeat sequence?
Great, thanks. So yes, what you say is true. And I think, one of the challenges that have come up with some of the PROTAC degraders that work through the proteasome is that you in fact get toxic fragments that remain. And autophagy is a little bit different than that, in that, first of all, we're targeting the primarily the fragments, although we do reduce full length. And so we don't see that as a problem. Let me just come back. And what I should have said first is we have been fastidious in testing against different CAG repeat lengths, both in our human fibroblast and iPSC-derived neurons, as well as in the animal models. And so we can hit everything from your, the smallest of 43 repeats, which is really just normal adult onset, to anywhere up to, I think we've tested 175 repeats. So the other thing I'll just tell you is that it's probably important that we hit the fragments, because that actually is seen to be one of the most toxic. And for Huntington, there's a missplicing that can happen that gives you the so-called exon 1, which is this 100 amino acid piece. And so if you can target that and get rid of it, which we can, and we've shown in animal studies, then I think that gives us, that's a huge advantage. So I share your concern, but I think that the pathway that we're working through allows us to avoid that. And I'll just comment, so I've actually targeted HSP90 and HSP70 in another lifetime. And the really big challenge there is that they have so many client proteins, and they have a role in both helping things fold and unfold. And so you've got a lot going on there. And so, yes, it's a central node, but you've hit a central node. And so that's always, that can be a double-edged sword. And in the case of the HSP90 inhibitors, even though we were selective for subtypes, we got as far as tox studies and found some unexpected tox. So yeah, thank you.
Beth, I just had you can educate me about this. So Phyllis and I are the Goofus and Gallon of pharma and med tech, right? And so I am the Goofus. I am the Goofus.
Nobody here old enough to know.
Okay, so just all right. So
I am.
All right, good. All right.
Highlights, right?
So when as a med tech guy, I see your timeline, I literally lose my breath, right? Because it seems so long. And what we're talking about is impact. I'm just curious, are there learnings along that timeline that allow for impact in the community?
Yes. So what I've done in terms of the timeline is, basically your average. So we know that we're not likely to have CMC issues with these molecules. We've scaled them up, not to tox levels, but that's been it's easy to make. 6 steps, no biggie. But what we know, and what I've done before, is we can get through the IND enabling studies in 9 months easily. And so we do have room to make this go a bit faster. Yes. Yeah.
Great job. Excellent presentation. Autophagy, not appreciated. My question is about the double-edged sword of autophagy and its role throughout the body. And, how do you see that?
Thanks so much. So we have a lot of data, which obviously I didn't have time to go through, but so our compound works through selective autophagy and not the autophagy you think about related to starvation and stress. And so the importance of that is, it is that we're not, and we've looked at this, and we've not affecting sort of the downside of the starvation and stress autophagy, which basically can lead to cell death, right? And then the other piece is we've done quite a bit of work showing that that we are able to change the shape of the mutant huntingtin for it to be captured by one very well-known cargo receptor in autophagy, namely P62. And so we can actually show that interaction in cells. So I think that we don't really have, a big issue. The other thing I'll tell you is that we often are doing 6, 14, or 21-day incubations with constant treatment with compound, and we don't see any untoward effects. Thanks.
So, I like the competitive landscape table. There's some really big players in this space. And so I'm wondering, are you already having discussions? Because if your compound has some of the advantages that it looks like over those in phase 1 and 2 already, with solid animal model data and even a little bit in humans, I would assume that the Roches and other are going to be really interested in this. And are you already having those discussions?
Yeah, thanks so much for this question, because yes, we are. So we're actually deep in due diligence with one pharma who's really quite interested, and and are starting discussions with a second, and a third big player wants to see non-GLP tox. So if we can get our non-GLP tox done, they would be very interested too, because they are very interested in HD.
So then exactly how does the funding from this challenge help advance you?
Specifically that, or the 5 million raise?
No, how does this challenge itself help your timeline and help your advancement?
Yeah. So this is what sometimes people think, drug discovery, you need millions and millions of dollars, but actually it's all done incrementally. And so the funding actually will help us get started on some of the synthesis and CMC so that we can move into the non-GLP tox. And so I think people who know, there's many, many things that have to be done. And so this really helps jumpstart us into the throes of the non-GLP tox. And that's huge. That's huge. As you know, it's been tough to raise money for therapeutics in neurodegeneration. And so people, like I said, sometimes people think, that's nothing, but it actually is quite a bit. And plus it keeps my people working to be able to actually generate that data. So that's what we would use it for.
Thank you, Dr. Hoffman. All right, last but not least, Neil Amin with SCULPTA Bio. Welcome, Neil.
Hello. All right. Let's see if my voice still works. It kind of does. I had a little bit of a cold before, but then I woke up yesterday and just shot voice. So I apologize if I was not very social yesterday. I wish I could have, but wanted to save my voice for today. So clickers here. So first off, it's exciting to be here. Exciting to hear about all the other companies and just the great energy around neurotranslation. So my name's Neil. I'm a physician and a scientist, and I am excited to tell you about our company, SCULPTA. We are sculpting the future of human brain health for patients with neurodegeneration, as well as for the aging population. So my background, as I mentioned, I'm a physician, a scientist, MD, PhD. I did my residency and postdoc at Stanford, became clinical assistant professor there. And, for a long time, really loved what I did in academia. Published lots of papers just in the last year, 2 papers in Nature, a first author paper in Science, and another first author paper in Cell Stem Cell. And probably from the outside, it seemed like that's what I'd be doing for my whole career. But when I first became a physician scientist, or wanted to become a physician scientist, I really believed in the idea of taking a really deep dive in basic mechanisms of biology and taking them to patients. And I lost faith that I could do that in my academic training path, and took the big leap 1 year ago, quitting my job completely, to doing something crazy of launching a company. I'm a first-time founder. I'm a solo founder. I never worked in biotech. And I was raising in the worst biotech market that we've seen. So clearly I'm a masochist. But really, I was just stunned to see how many people really believed in our vision, wanted to support neurotranslation. And we raised 2.6 million, oversubscribed round in a pre-seed from some really phenomenal investors. And that was just around 6 months ago. Now we have this fantastic team based mainly in San Francisco, but also with folks in Croatia and in the Netherlands as well. And so I don't really need to tell this crowd about the importance of brain of drugs, developing new drugs for the brain. But I do feel like there's a lot of excitement right now because there's growing appreciation of the population's only getting older, and neurodegeneration rates are only getting higher. The FDA has signaled that with all the availability of biomarkers, that they're willing to approve drugs based on these biomarkers, such as the approval of tofersen based upon biomarkers just alone. And I do think there's excitement with actually generating therapies for Alzheimer's disease, despite their shortcomings, that now there's opportunities to develop additional therapeutics on top of the ones already approved. So here at SCULPTA, we're very interested in neurodegeneration and the aging brain, and really finding therapeutic strategies that allow us to expand the opportunities of drug pathways and pathophysiology beyond what biotech and pharma are currently doing. And so what we know about disease pathophysiology and neurodegeneration is that over time, your brain accumulates different proteins. These signal and trigger lots of inflammation, and this will signal to neurons and eventually cause their death. And this really causes an immense amount of brain shrinkage over the lifespan. Protein aggregates can look like amyloid in Alzheimer's, synuclein in Parkinson's, or even just in aging. There's a protein called lipofuscin that just develops over, that builds up over time. And recently there anti-amyloid therapies have been approved that really effectively clear amyloid from the brain, but unfortunately the clinical benefit is rather modest. And despite the billions of dollars, truly billions of dollars spent on amyloid and tau over really decades, there's just limited clinical benefit and really a need for new directions in therapeutic avenues of really treating the disease. Also, the downsides of these antibody therapies is that there's increased risk of brain inflammation actually caused by the therapy itself, and also very frequent dosing. So when we looked at how can we really innovate in drug discovery through novel target pathways, it became really clear to us that the biggest drivers of Alzheimer's risk from really high confidence genetic genome-wide association studies are really landing in microglia. So these genes associated with Alzheimer's are really highly expressed in microglia. Some of them really almost specifically expressed in microglia. So what are these cell types? Oops, too far. There we go. So these are the brain immune cells. So even though there's been, I don't even know how many drugs that modulate the peripheral immune system, there have been no drugs ever approved that specifically target the brain immune system. I think it's a completely missed opportunity because these cells do an incredible amount. First of all, they clear protein aggregates from the brain. They modulate inflammation directly, and they also directly signal to neurons to protect them from damage. So with all of these sources of evidence pointing to microglial function, we really believe this is an exciting area of therapeutic development. So when I started the company, we really felt like the most powerful genetic tool that we have to get into new targets are oligonucleotide therapeutics. And it was brought up before, and it's great you guys already have a little bit of background on why they're exciting. But what inspired me and many other people is that back in 2016, when I was a graduate student, the drug Spinraza got approved for spinal muscular atrophy. This is a disease that's fatal for children. There's no treatment for it. And all of a sudden, you didn't need a stats background to see in that trial that most of the children were now surviving. It was remarkable and still is remarkable to this day that a new treatment paradigm could work so remarkably well. I think what makes this drug category especially powerful is that it is all the features of a futuristic drug. You can change the sequence. You get a new drug that targets a new part of the genome or transcriptome, but it has the same uptake into cells almost all the time. And this is now not new. I mean, there's now 20 approvals. I think the next decade we'll see that of all the genetic medicines, CRISPR, AAVs, that oligonucleotides are really the most clinically translatable. So where we come in is really expanding the use of this drug category to a wider range of targets and with different mechanisms, using our knowledge and of basic biology as well as our technology around single cell genomics. So our approach gets away from this paradigm of just trying to knock stuff down or increase levels, like say knock stuff down with CRISPR or increase levels with AAVs. We're actually doing what we call protein sculpting. This is where we actually change the structure and function of proteins in the brain. So how is this possible? This is through modulating alternative splicing with these antisense oligos. We can do this by tapping into existing differences and splicing that occur in the brain, or even create de novo variants. And just to give an example of how we can use this, is there are certain proteins that sit on the cell surface, a receptor with a transmembrane domain. We can actually skip the code for that transmembrane domain, and now this receptor is, instead of sitting on the cell surface signaling disease pathways, can actually be secreted, still binds its ligand, but no longer signals. These are the kinds of pathways that you can never imagine doing with a small molecule and are really specific to this pathway. So what gives us a very competitive edge is our technological background that combines GWAS data with really advanced, the most advanced single cell transcriptomics in splicing. So you might have seen around the news, we're kind of in the single cell genomics era, but 99.9% of that data is unusable for splicing discovery because it doesn't capture the full length of genes. We have built the largest database of single cell RNA splicing data by accessing roughly $30 million of data that's never been before analyzed in this way. This work is partially sponsored by AWS and Google. And we built the first human splicing atlas. We want to expand this to Alzheimer's, Parkinson's, and ALS because never before had been generated. This single cell splicing data has never been generated from any of these disorders ever. It's a really big opportunity for us to discover new targets. So from our platform, we already identified 2 targets that pharma is not going after, but we have built a very compelling case for it based upon our GWAS studies and our single cell splicing analysis. They both target microglial function in the brain for Alzheimer's with very high evidence through GWAS studies in Alzheimer's. We have in vitro evidence as well as a pathway to clinical trials because we actually have we're actually modulating a validated CSF biomarker that we didn't have to develop ourselves, but is really there that we can use to see if our drug's really affecting our pathway both in animal models as well as in humans. So I mentioned, we raised 2.6 million. It's really just enough to get us off the ground. We have very big ambitions of what we want to do. Our platform is really in the middle stages of development. We have really new technologies to actually capture single cell splicing from these disorders at a scale that other people have not through a new sequencing approach called BobSeq that'll be we'll have a limited release early next year. And our 2 assets have been identified from our platform. We're moving to iPSC models. So my maybe around 7 years at Stanford was all around developing the most advanced human brain organ models that anyone's seen. And we've had tons of papers on this. And really this gets us out of the kind of just the stuck place of trying to overly rely on rodent models for modeling human disease. One really important point here is that our second target doesn't exist in rodents. This happens all the time where human genetics lands in a gene or a gene pathway that just doesn't exist. And this is actually true with the drug Spinraza. SMN2 does not exist in rodents. If we limit ourselves to only gene targets that exist in rodents, we'd be much worse off. And so we are not shy about this, that we want to highly leverage our iPSC models and then move into non-human primates to advance these candidates. And so our goal is to raise a $10 million seed round to advance our platform and assets next year. And really importantly, it's been super exciting being here because I've seen how much Chris is he said today's his favorite day, but it's clearly the case that he's been working on this every other day of the year to build a community of people. Like I'm in the Bay Area, there's a great ecosystem, and that's really what it takes is an ecosystem of scientists, biotech entrepreneurs, clinicians, really everybody coming together. And it's very clear that he's brought everyone, here in the same way. And so what we're looking for is obviously always fundraising, and we're very excited to bring in new folks to into our company. But also, we have an exciting platform that can be applied across many different areas. We're really interested in generating new data sets from any kinds of clinical samples that you guys might have. I know there's a lot of also basic research going on in this area that that can lead to new drug targets. We already have one partnership with an academic center, helping them advance their target. That's what we're all about, is expanding the pool of target opportunities for patients. So thank you all. Appreciate it. And I'll take questions.
Okay, great presentation. Thank you for being here. I'm sorry you don't feel good. You couldn't tell during the presentation. Couldn't tell.
My voice just doesn't feel great.
So, question for you. You mentioned that amyloid and tau aren't moving the needle far enough, so you're looking at a separate mechanism. But you didn't really talk about what biomarkers you're using to actually show the activation. And while you reference the FDA biomarker approval pathologies, they're actually not in AD with the exception of amyloid. And even then, it's just accelerated. So question for you, I don't know if the CSF biomarker you mentioned is confidential or if you are thinking about a difference between biomarkers for engagement, target engagement versus tracking response to therapy.
Great questions. And because our innovation is in new target discovery, we can't say much about exactly what our targets are or the biomarkers which people will be able to figure out. But our approach is really considering this way that we want to have both a biomarker for target engagement, but really the real money, so to speak, of showing efficacies in validated biomarkers that have been well accepted as actually being related to Alzheimer's pathology. So that's what we need. I mean, that's the bar for really having a clinical trial. So that's what we're going for.
So you're really looking at the existing biomarkers that are validated with longitudinal cognitive assessment data.
Yes, that's right.
Okay.
Yeah.
Why do we need that?
Why do we need biomarkers?
That we already have.
Why do you need
No, So it's the same question you just asked. So we have tons of biomarkers, especially in the CSF, that are already validated or approved in clinical use. So I'm just what are you doing beyond that?
Mm-hmm. I mean, right now, what I think is exciting is that first of all, that there are now biomarkers that we don't necessarily need to develop both new assets and new biomarkers to get that's a lot to ask for. What we are actually developing is, and this is more on our platform side that I mentioned, is developing the first splicing biomarkers of disease. So we're really interested in collecting CSF, and you can actually collect some microglial cells in CSF and routinely in CSF. No one's ever used this for tracking any splicing drug that's ever been developed. I think it's a real opportunity for us with our single cell splicing analysis to actually develop wholly new types of biomarkers, actually great for actually target engagement. You can imagine if you're injecting a splice switching oligo, as we want to do, that you don't know if it's splice switching in living cells. And the opportunity to actually get cells from CSF is really a huge opportunity, and people just haven't done it before.
So just to clarify, to understand what the common nature of a lot of what your target discovery might look like, are these generally transmembrane molecules with internal signaling that themselves already go through endogenously alternative splicing. So there's up and down regulation and alternative splicing, and you're trying to nudge it toward that splicing that shuts down that signaling so that it stays in that state where the intracellular portion is not active.
Yeah, this is a great question. So the background here is that 90% of genes have alternative splicing. And when you really look at the data, it's like there's a little bit of noise everywhere. So there's a little bit of alternative splicing going on all the time. So it's a little hard to define like what is, happening or not. For one of our targets, roughly 10% happens at baseline. It's been like we can see it in actually primary human microglial samples and in IPS models, and everywhere we look, it's around 10%. And then for other target, it's actually that one, our second lead target is at 50%. We have other targets where, we have to be very selective where we go around, but some are actually completely novel in that like it's really low, extremely low levels. I think what's nice about if there is some baseline splicing, it actually gives us some indication on the biological side that it's actually easier to modulate those. If something's really zero, if there's something about the cellular machinery that's really trying to prevent anything from skipping that. The other interesting thing is that from evolutionary standpoint, there's actually a lot of reason that we can actually do this across almost every gene with multiple exons, because the exon shuffling hypothesis of evolution that most exons contain a domain of a protein. So it's not like there's randomly scattered, the sequence is randomly scattered. What that means is when we skip a exon, we're actually just removing one domain, the rest of the protein folds normally, and that allows really wide space for us to explore new mechanisms.
Yeah, just stay away from the bio weapon.
Oh,
right.
I hope not.
If you're tweaking what evolution is keeping you from ever alternatively splicing.
Yeah. Well, in the genetic medicine space, that's what we do.
Yes.
We play with genetics,
dual purpose.
But absolutely, yeah, we definitely want to be safe. Yeah, absolutely. Yeah.
Great job. What would the uses of funds be in the context of this this competition and in your current role?
Yes, so we're as you can tell, we're a very early stage company. We've just been closed around 6 months ago. That money really makes a big difference for us. First, I'll just say this competition makes a big difference for us. It's all about just getting out there and really appreciate that already. First, the money really helps us push how much we can do with our animal models. I think that's really that last bit that's elevates our impact and gets attention for us. And so that money really will be directed either towards, we already have quotes for non-human primate studies, will actually get us through our initial pilot testing there. And that would be really huge for us. Thanks. Thank you.
Round of applause for Neil and for all of our contestants for BioChallenge 2025. Okay, we are going to quickly shuttle our finalist judges to the Bioinnovation Center boardroom where they can begin deliberations. We'd like to ask our audience to hang for a moment. We'd like to invite last year's winner, Dr. Kaylin Hall, to come up and give a brief update about Inframuta, her company, and all the amazing things that have happened in the last 12 months.
Good afternoon, everyone. Like Chris said, I'm Kaylin. I'm the co-founder and CEO of Inframuta. And so first, I'd like to say thank you so much to Chris and the entire NOBIC staff for all their hard work to put this incredible event on. Thank you all so much for having me back. It's really fun to be back here in New Orleans again. I had a great time at dinner last night and getting to see everyone again and getting to know all this year's finalists. So for those who don't know, at Inframuta, we're tackling a grave growing problem of antibiotic resistance. This happens when antibiotics that we use to treat infections stop working on certain bacteria. And it's already linked to millions of deaths worldwide. And some projections claim that warn that it would claim over 10 million lives annually by 2050, rivaling heart disease and cancer. The timeline's urgent, and we need better solutions than the current trial and error approach that we use right now. So my co-founder Leo and I started working on this problem just 2 blocks away, right over here at Tulane School of Medicine. We wondered if we could transform the way that we tackle this problem and actually understand how bacteria evolve resistance and use that information to prevent it from emerging in the first place. So this went from a moonshot vision to a very personal problem and determination when Leo actually acquired a multi-drug resistant infection from a surgery, spent over 3 weeks in the ICU and almost died. And just a couple months later, we founded in Bermuda in January 2023. So since then, we've built a test that reads bacterial DNA and uses AI and predictive biomarkers to predict resistance, giving clinicians a heads up of what antibiotics would work now, and importantly, which could fail later. So that kind of unprecedented foresight could save lives at the bedside and support antibiotic stewardship at scale. It was incredibly special to return to New Orleans last year, where this all started to compete in the BioChallenge. The non-dilutive prize money that we won and all the connections that we made here directly catalyzed our initial prospective clinical validation studies at Ochsner. And also the prize money additionally filled out our funding round last year and allowed us to hire our key machine learning engineer to build our product. Since the BioChallenge, we've completed our first prospective clinical study with University of Pittsburgh Medical Center and also started 2 additional ones with Ochsner and also UPMC. We also have opened up lab space here at NOBIC, which will serve as our clinical lab headquarters when we launch the product. We've also solidified our partnerships with Ochsner and UPMC and are aiming to take the test all the way through implementation. We've recruited a top-tier lab director and chief medical officer, and we've also raised an additional $1 million in capital. I'm truly excited for all the finalists here and have been inspired by listening to everyone's presentations and innovations. I really resonated with Phyllis's message last night at dinner about New Orleans. The city and its people are truly resilient, gritty, and very special. One thing I'll add is this city and its ecosystem is one of the most friendly, collaborative, and eager to help I've ever seen. I really encourage all the finalists to take advantage of that, and no matter the outcome, take the time to make true connections here. And because everyone here really wants to genuinely help each other, and it will go a long way for who you meet here. So thank you all, and I wish all the finalists good luck.
Thank you. Thank you, Kaylin. Thank you, Kaylin. All right. So now it's time for us to have our audience vote for who their favorite contestant is. All of you have an equal opportunity to support one of these finalists with $10,000. So please pull up your phones. The audience award winner will win this really cool 3D printed light up trophy from Make Good NOLA, as well as $10,000 in cash, no strings attached. So please tell your friends and family who you want to vote for. Let's get as many votes as we possibly can up on the screen, and we'll let you know as soon as we possibly can. Okay, good afternoon, everybody. All right, we're going to wait a few more minutes for folks to come back in here, make sure all our judges are present. While we're waiting, please don't forget to tag us, connect with us online, take pictures on our glass wall in front of the brain, post them, and make sure that you sign up for our newsletter, our monthly newsletter at neworleansbio.com. And as I mentioned, right after this, we're going to be second lining down to Roosevelt Hotel Blue Room. I see some folks trickling back in. Okay, I'll go ahead and start my spiel while folks are coming back in here. Okay.
Are these the trophies?
Yeah.
These are the prizes. Wow.
Okay, guys. What an amazing show. What an amazing group of companies. What amazing variety of novel technologies dedicated to improve brain health. All of this outlines the unmet gaps that exist in the space, as well as the need to multiply the specialized expertise that we have here through advancing technology. The Bioinnovation Center chose neuro because it touches every family, every workforce, every community, and because convergence is happening across imaging, discovery platforms, neuromodulation, and cell biology that's giving founders real tools now. National stakeholders this week put it plainly for us. Neurotech spans biotech, medtech, digital health, consumer, longevity. This is the perfect frontier for a market like ours that lacks a niche and that is hungry, collaborative, and ready to lead. And we're doubling down. Because of the momentum we've built, because of the support of all these folks in the room, including Dr. Sid O'Brien, we're announcing that BioChallenge 2026 will again focus on brain health and neuroscience. This was just the beginning. We want more founders choosing this region, more co-development with industry, more investors who know that breakthrough biology can be built anywhere excellence is demanded. And here it's time to demand excellence, just like this little girl is demanding that her mom holds her. Yeah. Okay, so with that, we are going to announce our audience favorite winner. And I'd like to invite Andy Copland from the New Orleans Bio District up to make this announcement. And please feel free to share a little bit about Bio District while you're at it.
Little. Thank you, Chris, and good morning, everybody. I am going to get to open the envelope in just a second, but just thrilled to say that New Orleans is continuing to invest in partnership with our research universities, LSU, Tulane, Xavier, our health systems, LCMC and Ochsner, the Veterans Administration Medical Center. We have the assets to be able to have a strong innovation district focused on the life and biological sciences. Everybody here is part of that effort, and we're going to continue on a generational basis to make that investment to diversify our economy, to grow jobs, and to bring improvements to quality of life. And so you all are part of that first effort. The Bioinnovation Center has been leading the way, and we're going to continue to build momentum. So thank you all for being here this morning. And now I will open. So the winner of the audience favorite award is June Brain. Woo!
Thank you, guys.
Again, appreciate it. Okie dokie. Where's our, yeah, we need to hand out a fancy trophy to Samantha. So yeah, we get her, or maybe, no, yeah, we need to hand the audience, yeah. And we'll get a picture with that as well. Okay, for our third place winner, we'd like to invite Claire Durrett with Answer ALS to come to the stage to hand out the prize.
Thank you. This is really exciting. I get to hold a big check, although I don't get to take it home and deposit it to support our research. So without further ado, CENTRON BIO.
See how we're slightly winging it over here. All right. For the runner-up prize, for second place, we'd like to invite Bill Hines with Jones Walker, a champion of innovation in New Orleans and a champion of New Orleans in general. Thank you, Bill Hines, for all your support. We appreciate you very much.
All right, I won't do an infomercial. We'll just get right to it so you can go to lunch. The runner-up is origami.
All right, the moment that we've all been waiting for, I'd like to invite our presenting sponsor, Dr. Sid O'Brien, to the stage to announce the winner of BioChallenge 2025.
So this is a huge honor for me. Thank you for allowing me to do it. And I will say for all of you, excellent talks, excellent presentations, and it was very close. First place, June Brain.
Let me get this out of the way.
Yay. Oh, so you want her? I did. Trophy. Congratulations. Thank you. I would have picked her.
Thank you, everybody. That concludes this year's program and event. Please make your way to the lobby where we're going to gather to go straight down Canal Street to the Roosevelt.